Let's start at the very beginning. A very good place to start. When you're doing cellular respiration, you need C6H12O6 and 6O2. This becomes 6CO2 and 6H2O plus 36 ATP. All right, I'll make it easier. Listen. Do, 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 do. 1 is first glycolysis. 2, pyruvate oxidation. 3, Kreb cycle, that's 2 Kreb cycles. 4, electron transfer chain. That's all I got.
Glycolysis harnesses energy by breaking one glucose into 2 pyruvate molecules in the cytoplasm by the following steps. 2 ATP molecules are sacrificed to provide the energy required. The first ATP helps a phosphate join to the end of the glucose, creating glucose 6-phosphate. G6P rearranges to make fructose 6-phosphate, F6P. The second ATP helps a phosphate join to the other end of F6P, creating fructose 1,6-bisphosphate. These steps ultimately join a phosphate to the 6-carbon backbone. The backbone splits and forms glyceraldehyde 3-phosphate, which is also known as G3P, and dihydroxyacetone phosphate, DHAP. But then DHAP converts into 3GP. Now energy can be released. Each G3P sacrifices 1 hydrogen and 2 electrons to NAD+. NAD+ is reduced to nicotinamide adenine dinucleotide, NADH. While this happens, G3P joins with inorganic phosphate to form unstable 1,3-bisphosphoglycerate, also known as 1,3-BPG. 1,3-BPG gives a phosphate group to ADP, creating ATP and 3-phosphoglycerate, also known as 3PG. Each G3P leads to 1 ATP, so now we have 2 ATP molecules, which means the 2 that were originally sacrificed have been accounted for, and we now have a net of 0 ATP. This portion of glycolysis is called substrate level phosphorylation. 3PG rearranges into 2-phosphoglycerate, 2PG. 2PG gives 1 proton and 1 hydroxide ion. These combine and form water and phosphophenyl pyruvate. PEP is left. PEP is a very unstable molecule, and when it breaks down into pyruvate, it releases the energy required to transfer 1 phosphate group to ADP, creating ATP. Because there were 2 PEP molecules, 2 ATP molecules were created. Our net ATP is now 2.
This stage creates the acetyl coenzyme required for the next stage. The 2 pyruvate molecules move from the cytoplasm to the mitochondria. They pass through the outer and intermitochondrial membranes and enter the matrix with the help of a membrane carrier. Coenzyme A attaches to the pyruvate's acetyl group. This allows the pyruvate to be oxidized so NAD+ can be reduced to NADH. The remaining carboxyl group loses a hydrogen and is removed as the waste product CO2. Now all that is left is acetyl-CoA. When energy is needed, the acetyl-CoA enters the next stage to continue aerobic respiration. The Kreb cycle is a series of 8 reactions that transfers energy from organic molecules into usable energy for the cell. Krebs occurs in the matrix of the mitochondria. One glucose molecule must undergo Kreb cycle twice. This cycle releases CO2 as waste, and a specific enzyme catalyzes each reaction. The product of pyruvate oxidation, called acetyl-CoA, reacts with oxaloacetate to form citrate.
Yay!
Citrate undergoes isomerization and creates isocitrate. A redox reaction oxidizes the isocitrate and becomes alpha-ketoglutarate, which also releases CO2. An NAD+ ion reduces to become an NADH energy coenzyme. Alpha-ketoglutarate binds with CoA and creates succinyl-CoA. NAD+ is oxidized and becomes NADH. CO2 is also removed in this stage. Succinyl-CoA sheds its CoA through a series of phosphorylations. This series occurs as phosphate displaces CoA. Guanosine diphosphate, GDP, is phosphorylated to become GTP, and the phosphate group transfers from the ADP to form ATP. After these phosphorylations, succinyl-CoA becomes succinate. The FAD ion, found in the matrix, oxidizes succinate and creates the energy coenzyme FADH2. Succinate then becomes fumarate. Water is added to create malate. And finally, malate undergoes a redox reaction involving NAD+, producing NADH, and oxaloacetate, and this completes the Krebs cycle.
Welcome to Electron Transport Chain. The purpose of ETC is to make ATP through oxidative phosphorylation. ETC also occurs in the inner mitochondrial membrane. Chemiosmosis is ATP production in cellular respiration. This is the movement of ions from an area of high concentration to low concentration through a membrane protein, which results in the release of H+, which then makes ATP. Another important term is mobile electron carriers. Now, these mobile electron carriers carry the electrons through each protein in a mitochondrial membrane. And finally, electron carriers are ubiquinine and cytochrome C, and they carry electrons through the oxidation of glucose. There are 7 major components of ETC. One of them is complex 1, which is NADH dehydrogenase. The second component is complex 2, succinate dehydrogenase. Third component is complex 3, which is cytochrome B, which is the C1 complex. Fourth component is complex 4, cytochrome oxidase. Then, of course, fifth component is oxygen. Sixth component is ubiquinine. And the final 7th component is cytochrome C. Here's how ETC works. To begin, we start with an NADH molecule. Now the hydrogen on the NADH molecule goes into the first protein, and the electron goes into the protein and continues through with the help of ubiquinine, whereas the proton goes up into the intermembrane space. The electron then passes with the help of ubiquinine into complex 3, where then cytochrome C helps the electron move to complex 4. This whole time, the protons are following the electrons because they are attracted to each other. However, the protons are in the intermembrane space and the electrons are traveling through the proteins. Then FADH2 enters into complex 2 because you can't have the same ion in the same protein. So we go into complex 2, then move with the help of ubiquinine into complex 3, then with cytochrome C into complex 4. Then the remaining hydrogens go into ATP synthase, or they're like sucked into a vacuum, and they come out and the proton goes with ADP and makes ATP, which is energy. And now we have finally reached the overall goal of ETC, which is to create an electrochemical gradient to power ATP synthesis. Oxidative phosphorylation! Woohoo! So long, farewell, and now I must leave. You know the steps of how a cell does breathe.
Well,